Okay, thank you. What a nice idea these set of talks and seminars are, and so I'm really happy to participate. I don't have a patient, as I mentioned, but it's a sort of a very clinical application of some of the work that we're involved in, and it's got implications for all kinds of ethical and clinical utility applications that we can talk about as we go through. First of all, my financial disclosure. These are the sources of funding that I receive. I used to get royalties from Guilford Press, but nobody buys the book anymore. So if you want to contribute to that'd be great. Okay, so I'm going to talk a little bit about what do we mean by personalized medicine. And I want to very quickly sort of summarize in just a few slides some of the progress that we've made in identifying genetic variants in autism spectrum disorders. I'm going to pose the question whether we're ready for genetic testing as an application of personalized medicine in ASD, and I want to illustrate some challenges that I think are going to be quite difficult to overcome, particularly with our current genetic paradigms. And if you can't wait for the end of the story, I can tell you that we're not ready for personalized medicine, but it's happening anyway, so we better get used to it, and we better start thinking about some of the complexities and some of the challenges that are involved. So first of all, what do I mean by the concept of personalized medicine? It really is the use of genomic information to inform healthcare decisions. So you're taking information from an individual's genome, from their genetic material, and you're using that unique information to personalize the healthcare that they're receiving. So the genomic profile is really obtained from their DNA, and it's used to inform prediction, to predict an individual's status with respect to risk, to diagnosis, to the course that they might follow, or to their response to a particular treatment intervention. And it's really driven by the concept of disease heterogeneity. That is, that there are, even though there's a single diagnosis or there's a class of individuals that might share a particular diagnosis, whether it's autism spectrum disorder or cardiovascular disease or diabetes or whatever, no two patients with that diagnosis are the same. And actually, this is a concept that was first sort of promoted by Sir William Osler in his famous textbook. You know, Sir William Osler lived in Dundas, eh? And you can still, his house is still there where he lived. It's an absolutely gorgeous house. It's used as a bed and breakfast now, and it's so you can go and visit it. It's quite lovely. So let me just illustrate about how personalized medicine is supposed to work. And there's a really nice article in the Lancet 2010 by a group from Stanford that's first author is Ashley. And what they did was that they took the entire DNA sequence of a healthy 40-year-old male. He was totally healthy, no health problems, but he did have a family history of sudden death, and there was a family history of cardiovascular disease. And sequenced his entire genome. They looked for genetic variants that might be associated with different Mendelian diseases, looked for novel mutations in his DNA, looked at genetic variants that are relevant to pharmacogenetics, and SNPs, genetic markers. A SNP is a genetic marker that's associated with various common diseases. And what's really cool about this particular approach is that they calculated something called the likelihood ratio. So the pre-test probability, the likelihood ratio, and the post-test probability. And I'll just illustrate that in a second. So let's assume, for example, that the pre-test probability of a disease is X. That is, in the general population or in a particular sample, the pretest probability can be identified. So let's take, for example, that in 40-year-old males, the pretest probability for something like depression is 10%. And then you can calculate the likelihood ratio from the sensitivity and specificity of a particular genetic marker. Actually, it can be any diagnostic test, but in our context, the diagnostic test is a genetic marker or a group of markers, and you can calculate the sensitivity and the specificity of those genetic markers for a particular outcome. And let's say I've got some genetic markers and I have a certain sensitivity and specificity, and from that I can calculate the likelihood ratio. The likelihood ratio means how much more likely is it that the individual will have the outcome given a positive test relative to having the outcome given a negative test. So it takes the sensitivity and specificity of the test and then calculates how much more likely you're going to be to have the outcome given a positive test on the result. So the post-test probability, so the probability you're going to have the disorder is basically the pretest probability times the likelihood ratio. So if the pretest probability is 10%, the likelihood ratio is, let's pretend, 5. You're 5 times more likely to have depression if you're positive on this genetic marker than if you're negative on this genetic marker. You give the test. If you're positive, the post-test probability is going to be 50% that you're going to have that 40-year-old males are going to be depressed, for example. 40-year-old males with this pattern of genetic markers that are positive, 50%, the post-test probability is going to be 50% that those individuals will have depression. So this is the list of post-test probabilities of this 40-year-old male given, so here are all the different outcomes that they looked at, and here is the post-test probability, and here's the estimate of the post-test probability for this particular individual. And they sort of calculated it in this sort of fashion. The length of this refers to the sort of the confidence intervals around the post-test probability. So what they were able to find is that amongst his pattern of DNA sequence, there were several genetic variants that were strongly associated with cardiovascular disease and with sudden death. So this individual was at increased risk of sudden death and developing cardiovascular disease. Remember, some variants that they looked for had pharmacogenetic properties, and so the pattern of his DNA also indicated that he had some variants that were associated with a beneficial response to statins and a reduced risk for other side effects associated with statins, like skeletal myopathy. A number of variants were identified that were associated with Mendelian diseases, hemochromatosis, but in this case there was no family history of that. So they decided to treat this individual, who was, remember, asymptomatic, with statins to prevent the risk of cardiovascular disease and sudden death, and that he needed close monitoring for the possible development of hemochromatosis, because he had these variants that were associated with a particular Mendelian disease. So you can see how this could actually be very, very useful. You could develop with completely asymptomatic, or individuals who were mildly symptomatic, or even individuals who had a disease, and you were trying to make treatment decisions. You could use their genetic profile, if you had the information, in order to determine what drugs they might respond to, what disorder might occur, what course they might experience over the course of their disease, etc., etc. So now I'm going to ask the question is whether it's possible to develop a personalized approach in autism spectrum disorder. So So I'm just going to take a step back and make sure that we're all on the same page with autism spectrum disorder. You know, this is I'll just talk a little bit about what ASD is. It's a severe disorder of childhood, onset prior to 3 years of age. It's associated with difficulties in social interaction, difficulties in communication, and this pattern and preference for doing the same thing over and over again, or developing very restricted interests that are a little bit unusual, like, I don't know, stamp collection, or, you know, hockey statistics, or, you know, things like that. It's really the intensity of the interest that is often unusual. Boys outnumber girls by about 4 to 1. Prevalence is about 1% in the population. So there's been a lot of interest in the genetic epidemiology of autism spectrum disorder. And one of the things that's driven this particular interest is whether we can develop genetic variants or have an understanding of different genetic variants that would be useful in early diagnosis, in treatment planning, in helping us to identify what the cause of the disorder is, et cetera, et cetera. So in this case, when we're asking about personalized medicine, I'm going to ask whether we can develop a genetic panel and a set of genetic variants that could be useful in personalized medicine by calculating these likelihood ratios. Now, we know that ASD is a genetic disorder, so it runs in families. If there's one individual in the family that has autism, the probability that there'll be a sibling that will also be affected is about 5 to 9%. The most recent data indicate that actually if the firstborn has autism, the probability that a secondborn will have autism is more like 20%. So if you're just looking at the secondborn individuals, the probability is a bit higher. That only tells us that ASD runs in families, doesn't tell us whether it's genetic or not. We need twin studies to be able to disentangle that. We compare the concordance rate in monozygous twins versus dizygous twins, in which the twins, one of whom has autism. And a number of different twin studies have been done, all of them showing that the concordance rate in monozygous twins who are genetically identical is much higher than in dizygous twins. So heritability, upwards of 90%. Heritability refers to the amount of phenotypic variance that can be accounted for by genetic liability. So it looks like it's a highly genetic disorder. It's probably the most genetic of the neuropsychiatric disorders. So is it possible that we can identify a set of genetic variants that could be used in a personalized medicine kind of approach? Now, the issue is that the Ashley paper, the one in the Lancet on that 40-year-old male, all that information about those genetic variants were obtained through what are called GWAS, Genome-Wide Association Studies. So Genome-Wide Association Studies, just to sort of go over that really, really quickly, it's a simple case-control design. You take X number of individuals with a disorder, and you take controls without the disorder. And it's what we call hypothesis-free. So you're not looking at any particular genes, you're covering the entire genome. So you'd have 1 million genetic markers to cover the entire genome. And all you're really asking is, are there certain genetic markers in the cases that are more common than you see in controls? It's so simple, it's so brilliant. It's one of those simple, brilliant ideas that you think, well, why didn't anybody think of that so long? Well, the reason they didn't think of that was because there was no way anybody could get 1 million genetic markers to cover the genome from chromosome 1 all the way down to the bottom of the X chromosome. The trick is, with these genome-wide association studies, is because you have 1 million genetic markers, you've got multiple testing, so you have to correct for multiple testing. So the p-value, the significance level, in genome-wide association studies is 10 to the minus 8th. None of this 10 to the 0.05 or p less than 0.05. We should be so lucky. A significant result in a genome-wide association study is 10 to the minus 8th. And then in these disorders with cardiovascular disease and diabetes and Crohn's disease, the ones that have been really successful, then you can develop a number of different markers. I think in diabetes, for example, 18 different genetic markers have been identified that are associated with diabetes from GWAS. So what happens in autism, in GWAS and autism? So I was lucky enough to be part of a consortium called the Autism Genome Project, which was 13 different groups from around the world. We had a sample of 1,500 families altogether and thousands and thousands of controls. We pooled all our families from all these different groups together. We had a common genotyping platform. We had a common phenotyping platform. So the clinical measures were identical. And we looked to see, are there certain markers that are more common relative in the cases of autism than in the controls. And here's the result in a single slide. So just to sort of orient you, on the bottom here, you've got all the different chromosomes, including the sex chromosomes. And here you've got the p-value. So it's fixed, it's turned around so that if you get a p-value up here, which is 10 to the minus 8th, then you've got a significant result. Each of these dots represents a genetic marker, and this is what's called a Manhattan plot. And this is for cases of autism of all ancestry with an autism diagnosis looking at an additive model. This is a broader kind of diagnosis, so instead of autism, we have the broader category of autism spectrum disorder. And the bottom line is that there's only one little dot that makes it even close to the 10 to the minus 8th value. It's called macro D2. It's an interesting, the marker is in an interesting gene. The macro D2 gene is a brain-expressed gene. So it's not inconceivable that this is a gene that might be involved in the etiology of the disorder. But you know what? There ain't anything else that comes really even close to the 10 to the minus 8th. So that was really, really disappointing. So we concluded at the end of that GWAS study that there's maybe one genetic marker that barely reaches, it's sort of approaching, hanging on by its fingertips to the 10 to the minus 8th value. We then looked at other subtypes of autism spectrum disorder. We looked at quantitative traits. None of those reached genome-wide significance. Again, you have to correct for multiple testing. There have been 3 other genome-wide association studies published. None of them have replicated the results of any other. So each GWAS gets 1 or 2 SNPs that are close to significance, but none of the other studies can replicate that. So the bottom line is that from GWAS, we haven't really been able to identify any genetic markers that are significantly associated with the disorder. Now, why is that? Well, you have to understand that there's sort of 2 competing paradigms in complex genetic disorders, human disorders, that are fighting it out, as it were, with respect to which sort of makes the most sense. There's the common disease, common variant paradigm, and then there's the common disease, rare variant paradigm. And GWAS, genome-wide association studies, are really designed to capture the common disease, common variant hypothesis. So along the bottom here, you've got an idea about the frequency of the genetic variant, and along the y-axis, you've got the relative risk of a particular genetic variant. That is, how strongly is that variant associated with the particular outcome. And GWAS, or the common disease common variant hypothesis, proposes that there are a lot of common genes associated with diabetes, cardiovascular disease, obesity, height, general cognitive ability, but their relative risk is low. So the more common they are, the relative risk they have for the outcome is pretty low. It's when they aggregate together that you get the disease. The rare variant model says, well, okay, we've got rare variants that are less common, but we believe that they're associated with a higher relative risk. So they have a stronger effect even though they may be rare and less common. And it's sort of these two paradigms that are part of what we call the allelic spectrum that are at the opposing end of what seems to be driving human genetics. So, GWAS didn't come up with anything, so let's hypothesize that there are rare variants associated with the disorder. And you know what? The rare variant model makes a lot of sense. And the main reason for that is that people with autism don't reproduce. They don't have children. So it's what we call a genetically lethal disorder. If that were true, you would expect the prevalence of the disorder to be going down over time because the genes taken out of the gene pool every time somebody with autism dies and doesn't have children. But the prevalence of the disorder is not going down. If anything, the prevalence of the disorder is going up. It's probably stable, but it's certainly not going down. So every time somebody with autism dies and doesn't pass on their children, there has to be a new case, right, that comes into the population to contribute their susceptibility genes to the gene pool. And that's the idea that there are de novo genetic variants, that there are spontaneous, let's not call them mutations yet, but there are spontaneous genetic variants that arise de novo in the sperm, in the egg, and that then causes susceptibility to a particular disorder. And if they're de novo, they've got to be very rare in the general population because they arose in my genes, in my sperm and egg, right? And not in, they won't have arisen in anybody else's. So it's got to be rare. So the hypothesis here is that ASD is caused by a lot of rare genetic variants. And one example of a rare genetic variant is something that we called copy number variants. So I'm not going to go into a lot of detail about this, but you know, we all have 2 copies of every gene, right? Even I learned that at McMaster Medical School. And copy number variants occur when you only have 1 copy of a gene, you're missing a copy of a gene, or you have an extra copy of a gene, there's a duplication of a particular gene. So our DNA, you know, is really messy. It's full of holes. It's like Swiss cheese. There are bits of DNA that are missing. There's extra bits of DNA that are tacked on there. There are all kinds of deletions and duplications. And if they're big enough, over 1,000 base pairs, that's called a copy number variant. And they can be rare or they can be common. They can be inherited. I can have a de novo copy number variant and pass it on to my children. And if it's benign, it won't, you know, that my kids will pass it on to their children if they ever have any, and so forth and so on. So you can get inherited copy number variants, but if the copy number variant has an effect on human disease, if it's a risk factor for a disease, then it tends to be more and more rare, especially the extent to which it leads to reduced fertility. So this is just an illustration of what copy number variants look like. So this is a reference genome. This is a reference chromosome, for example. And this might be the chromosome of an individual with autism. And you can see that this section of the chromosome in here has been deleted. It's not there. Or you can have a duplication. So here's the reference chromosome. You can have a duplication of that section of the chromosome in an individual with autism. So you have deletions and duplications. And the argument is that if you have these deletions and duplications in genes that are expressed in brain, and they affect the function of that gene, because not all copy number variants affect gene function. If it affects the function of that gene, maybe that gene is involved in the etiology of autism. Well, so then we went back to our sample of 1,500 families, and we looked for copy number variants, particularly in in brain expressed genes, and genes that were associated in the literature with autism or with intellectual disability. And the bottom line is that in about 7% of the individuals with autism, we found copy number variants relative to about 3.5% in controls. And that was significant at a genome-wide level. So amongst this sort of catalog of about 300 genes, we were able to find copy number variants in some of those brain-expressed genes in about 7% of the individuals with ASD relative to 3.5% of the controls. So copy number variants appeared to be something that had some traction. And since the publication of that paper, this whole area of copy number variants in ASD has exploded. And people are identifying copy number variants in all kinds of different genes that are related to the brain. Each copy number variant is quite rare, less than 1% of the cases, but because there's so many of them, they're accumulating more and more and more. Now, I'm not going to spend a lot of time on this, but this is Karen can maybe explain this a little bit better. Maybe he'll talk about this a little bit more. But this is just an illustration of the axon, the dendritic spine. And what's been really interesting, that a lot of these genes here are actually functioning at the level of the dendrite and the axon, in this synaptic space here, or in this part of the neuron related to synaptic development or axonal development in one way or another. So although we've got hundreds of different genes, we've got a smaller number of pathways in which those genes operate, in which those genes interact with each other. So the story's beginning to kind of make sense, and we're kind of beginning to make progress. Okay, so let me bring it back to the personalized medicine. So it doesn't look like common variants play a role in the etiology of ASD, but it does look like rare variants. And would it be possible for us to develop a panel of rare genetic variants and use that for diagnostic testing in some way? Some of you may know that this is already being done by different companies in the United States. You know, you can send away your DNA to 23andMe or some of these other companies, and they'll tell you if you carry any of these rare copy number variants that are going to increase your risk for autism spectrum disorder. So, in what context are we going to be able to use genetic testing in terms of its clinical utility? And you know what? There's one area where it would be possible, where it kind of makes a lot of sense. The average age of diagnosis of kids with ASD is between 4 and 5 years of age. Parents first become concerned with their kids by around 18 months of age. So there's this huge window between 18 months and getting the diagnosis at 4 and 5. And what's really tragic is that parents have a hard time getting that diagnosis made, and it's that time period when the kids are most susceptible to early intervention. Early intervention can make a big difference, but only if you get there by the time you're 6 or 7. So if you're not getting the diagnosis till you're 5, you don't have a lot of opportunity for early intervention, as opposed to getting a diagnosis at 18 months. Then you've got a lot more early intervention to be able to participate in. That's going to improve your outcome a great deal. So is it possible that we can use genetic testing to lower the age of diagnosis so that kids can get enrolled in an intervention program much sooner? So let's have some fun with the numbers, right? Love numbers. Let's assume we've got a clinic where there's a whole bunch of kids that have developmental problems of one sort or another, non-specific. Let's say the pretest probability that kids in that sample are going to develop autism is about 10%. So that's the pretest probability. That's the prevalence of autism in this clinic of non-specific developmental disorders. Let's assume, remember I had 7% and 3.5%. I'm going to change the numbers a little bit just for fun. Let's say it's 6% in the cases of autism, 2% in control. That's sensitivity and specificity. So my likelihood ratio is 3. So that is, likelihood ratio for a positive test is sensitivity over 1 minus specificity, 6% and 98%. That gives you 3. So the pretest probability is 10%. If the likelihood ratio is 3, the post-test probability is 30%. The post-test probability for a positive test. So in these settings, a positive test and with a high pretest probability, you know what? The diagnostic panel works pretty well. Going from 10% to 30% is a pretty good confidence that it's worthwhile giving a kid a diagnosis or really seriously investigating the probability. Now, what about a negative test? So what's the likelihood ratio of a negative test? Well, here it's 0.96, using the same numbers of sensitivity and specificity. The pretest probability is 10%. But if I get a negative test, I'm only reducing the probability that that child has autism to 9.6%. You with me? So a positive test increases the probability from 10 to 30. A negative test is useless. It's only reducing the probability from 10 to 9.6%. So a negative test actually means nothing. And because the variants are rare, virtually everybody's going to get a negative test, right? Because I'm only getting 6% of the cases that have a positive test. 94% of the cases have a negative test. So a negative test is absolutely useless. But a positive test will be helpful, but it's going to be rare. It's not going to be very common. So, bottom line, is ASD ready for personalized medicine? Only if the test for a copy number variant or for a particular genetic variant under our current standards, if the test is positive, and only in samples with a high pretest probability. Okay, so I've been pretty negative about genetic testing and all this genetic research. And you know what? A lot of people are pretty negative about personalized medicine used in diagnostic circumstances. It does, however, seem that there's more promise when it comes to therapeutics. Now, a really important message is there are no medicines that we have that are directed at the core deficits of autism spectrum disorder. So we don't have any drugs that affect social communication and repetitive behaviors. But what's really exciting is that people have taken these genes that I've identified, developed knockout mice, for example, and have administered medication where they can reverse some of the cognitive deficits in these mouse models. So I'm going to stop at that point, and I'm going to let Karen take over from here. I'm just going to recognize my colleagues in Canadian. So we're known as the Canadian Autism Genetics, short form Canadian. Pretty good, eh? Huh? At Sick Kids, Steve Shearer is a huge leader in the project, really one of the first scientists in the world to really study copy number variants and point out their importance in human human disease. Do I take questions now, or do we do the whole thing?
We can take questions now. We don't record your questions, but it's
Okay.